DNA replication is a super important cellular process that happens during interphase. It takes place in the nucleus for eukaryotic cells. Before we begin the process, let's introduce our key players. Nucleotides, the building blocks of DNA, are each made up of one nitrogenous base. The four bases are adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, and cytosine and thymine are pyrimidines. Nucleotides are also made up of the pentose sugar deoxyribose and a phosphate group, and together they make the backbone of DNA. The process of DNA replication is run by a few enzymes. The main ones are DNA helicase, the unzipping enzyme, DNA polymerase, the builder enzyme, and DNA ligase, the gluer enzyme. Here we have our original double-stranded DNA about to undergo replication. Firstly, helicase comes in and breaks the hydrogen bonds between the complementary base pairs. This separates the DNA into two separate strands. The top strand, which is the leading strand, runs 5' to 3'. DNA polymerase can only build in the direction of 5' to 3'. Now, DNA polymerase comes in and joins the bases with their complementary base pairs through hydrogen bonding, and the adjacent nucleotides also join together. It also builds the sugar-phosphate backbone as it moves along. The leading strand is easy for DNA polymerase because it runs 5' to 3'. The lagging strand on the bottom runs 3' to 5'. DNA polymerase, therefore, must build from the opposite direction. It has to keep catching up to where the DNA is unwinding, and you can see this is why it is called the lagging strand. This process creates these fragmented sections of nucleotides called the Okazaki fragments. Now, ligase comes in and seals the gaps between the Okazaki fragments. The final product of DNA replication is two identical strands of DNA, which are also identical to the original parent strand. DNA replication is considered semi-conservative. This is because the two DNA strands are each made up of one original strand of DNA and one newly created strand. Thank you so much for watching my stop-motion video on DNA replication.